more about ACP, because sometimes you do get students from here from an exchange program to come and work with our colleagues on nematodes. So this is just going to give you a few highlights about the institution. A few facts: we're a center of excellence in Africa for research and capacity building. And Laura was talking a lot about capacity building. This is why we have that kind of exchange program where we have students come from Kenya this way or East Africa this way. And then we also have students from here. And then they have that kind of interaction for a training course. The other thing about the institution is that it's an intergovernmental organization signed by 13 countries worldwide, about 500 staff members with several contracted workers. And on average, annually, we do have about 150 to 180 graduate students, MScs and PhDs, and several interns, undergraduate interns from the local universities. We also do get students come from various parts of the world, apart from our resident PhDs and MScs who come through a sandwich program. Now, we have a unique history because it was started by an African from Kenya, and most of our scientists come from all over the world, but there's also that dominance because we train a lot of African students, and our capacity building program in terms of postdocs and so on. And then those who become early career scientists also become from Africa. So we are very unique in that respect. So you look at our beautiful buildings over there. When you get into Nairobi, please pay us a visit and interact with our. You'll always find somebody who potentially you can collaborate with. So don't spend all your time in safaris looking at animals, but visit ECP as well. Okay, and then how do we work? Now, ECP has three thematic areas. One focused on human health. The other one is agriculture, which is plant and animal health. And then environmental health. You can clearly see highlights of the areas we work in and the human health, malaria, leishmania, zoonotic disease. But these are the vectors that we focus on. Okay. And then again, when it comes to agriculture, it's the same thing. We work on the pests that are associated with it. And we have to focus, we can't work on everything. We have to focus our work on certain areas like staple foods like maize, sorghum. Then we come to horticultural crops, which is going to be one of the areas I'll talk about. Tomatoes, potatoes. Then you have migrant pests like locusts. Fall armyworm is now a big problem. Drosophila Suzuki is also a big problem now. And the animal health, we actually work on tsetse and ticks and biting flies. And then environmental health, when we're doing all this, we have to actually ensure that whatever we come up with is environmentally sound. So biodiversity conservation is very important to us. Beneficial insects are also very important to us. So bee health, we have a huge diversity of that in Africa, and we should be taking care of them. And then also we have commercial insects like silkworm and so on. Then applied bioprospecting is to look for new biocontrol agents, or maybe what I would say biological control methods, which includes looking for new secondary metabolites for pest control, and also the influence of climate change on all these interactions. Cutting across all this is capacity building. We have to have students to do that. That's why we have about 150 to about 180 students at a time, and early career scientists doing this job with that. And these programs, we do have what is called the African Regional Postgraduate Program in Insect Science. There's another one which is called Dissertation Research Internship Program, DRIP. And these are this PASET, which is an area where we partner with those who are in engineering, science, and technology. And then there's a regional scholarship, innovation fund. These are all capacity programs within ECP. which is really very helpful. But the question is that if we do have all these programs, how do we get the job done? And getting the job done, ECP is established units, and these units are 8, where the underlying science is actually carried out. And I've been fortunate enough for almost 2 decades to spearhead the research at the Behavioral and Chemical Ecology unit, where now I'm an emeritus fellow. But other than that, I think I've had almost 3 decades working within this unit. Now, the interesting thing now, it's about going below ground. Having talked about behavioral and chemical ecology, we've been doing that always above ground, looking at interactions between insects, plants, insects between insects, and look at message-bearing chemicals, which we call semiochemicals. Okay, it allows us to identify chemicals that are, mediate reproduction, like sex attraction, or finding a host, what you call pheromones, or maybe repellents, pheromones, and so on. We've been doing all that above ground. And like I said, our informal conversations with our collaborators from ITA guided us to begin to look below ground, because the foundation is always very important, and that could contribute to why certain plants may be very vulnerable to insects. So how does chemical ecology now come in? Because it's all about communication between organisms. So the issue about nematodes, you don't really need to be a nematologist, but you have to know that you're working with this specific nematode. But as far as the basic science, it's the same. So to get into this, we actually got 2 of our very shining stars. 2 ladies here. This is Ruth Keika from Kenya, to look at root knot nematode interactions with various plants. And our model plant here is actually tomato, which is a Solanaceae. And then the focus is on Melanochyme incognita, which is very common in Kenya, and of course, the sister one, which is Melanochyme javanica. She's been working on both these 2 species, and we actually find that there isn't any real species specificity regarding these 2. There's, interchangeable in terms of their responses or interactions with various host plants. The other one is potato cyst nematode, which is a new invasive species in Kenya. And again, another shining star. She came as an MSc student to work with us. Juliet Ochola, and Juliet is currently now a PhD student at North Carolina State University. And she did a brilliant job. So these are 2 shining stars within our nematology team. And so where does chemical ecology come in when we talk about root knot nematodes, interaction with tomato, and then potato cyst nematode interaction with potato? We can only identify the areas where chemical ecology comes in and then interrupt or disrupt that kind of interaction only if we understand the life cycle. So the first thing we all had to learn was the life cycle of plant parasitic nematodes. And clearly you can see with the root knot nematode and cyst nematode, we all know that the root knot nematode can easily hatch even in water, but then the cyst nematode needs a chemical stimulant, which is actually from the root exudates of the host plant. And that is the very reason why it can remain in the soil for over 20 to 30 years until it gets the right signal, particularly in the case of potato cyst nematode, potato root exudate. So that's where the chemical stimulant comes from, a Solanaceae plant, like tomato or potatoes, for the cyst nematodes to hatch, but not for the root knot nematodes. But then once the hatching occurs, then you now have the motile stage, the one that now has to seek to look for the roots, to penetrate the roots, and that is the J2s, which I suppose those who are plant pathologists, you know a lot more about this and so on. But what happens is that it's the J2 now, that is the motile one that has to look for it. And then again, it's based on chemical signal, okay? So as far as we're concerned, as chemical ecologists, This is where the weak link is, because once they've penetrated the roots, they actually create a sink where most of the nutrients ends up diverted to them for their own development, and that deprives the plants from, growth. And so once that occurs, you can actually now get the plant parasitic nematode complete the life cycle. But if you disrupt it, then that is not going to happen. In the absence of the disruption, this entire process, something is missing here. The entire process goes through, and then eventually you end up with this kind of damage. And cause obviously a clear indication of infection and poor harvest in the case for smallholder farmers. Now, our target group is always smallholder farmers. So whatever we do, we have to make it simple to ensure that it can be adopted by our stakeholders. So the first question, after understanding the life cycle and where we've identified the weak link, where chemistry is concerned, is how do we now go about this? We asked the question, for the case of Ruth, do olfactory cues mediate RKN host interaction? Basically asking, are there message-bearing chemicals that they detect? And if we can understand this, then it means that we have an opportunity now to disrupt it. We first have to understand the problem, then we can come in with a solution. And the very first thing in chemical ecology is to design a very sensitive bioassay. If the bioassay doesn't work, the chemistry is going to be wrong. If you have the right bioassay, then the chemistry is now going to follow. There's a likelihood that you are going to get it right. So the behavior drives the chemistry and not the other way around. So the very first thing was to design a bioassay. I said just a two choice of factometer, with sand in there. And like I said, it's not the kind of bioassay where you're working with an insect and you have a glass tube and you can actually see the insect moving. This is below ground, and that is where the challenge is and so on. So we came out with these two choice of factometer, and you can actually, one side put the plant, and then the other, you can actually even infect the plant with nematodes, specific number of nematodes. So you can play around with it. You can actually put two plants in there to simulate even intercropping to find out whether it disrupts and do all kinds of things. You can even expand this to a 4 choice of factometer, a 6 choice of to play around with so many plants. So this was the very first thing that we had to design. And here in the case of tomato with sand, what this means, NI indicates that that's non-infected plant, tomato roots. You clearly see the response is very high, almost about 80% response compared to sand. But then when you do have 2 days post-infection, look at the red, there's avoidance of the tomato plant. So something must be happening in terms of the chemical communication below ground. But then 7 days, a week later, now there's a preference again for the tomato plant. So then what potentially happens is that when you have the intact plant, the message is that this is great. Let me actually now infect. The moment you have infection, the plant has to respond to defend itself. The physiological response is going to be very different. And the root knot nematode is able to detect it to avoid it. But then 7 days later, the plant recovers. And the root knot nematodes are able to tell that the defense compounds potentially are no longer in high concentrations. They can infect again. Okay. So this information was really very useful for us to look at the chemistry. But before then, we need to validate this information. And the validation is now to look at when you have intact tomato plants and now you have 2-day post-infection and 7-day post-infection, you can actually make that kind of comparison. That's what we did. So here you look at the blue, when it's non-infected versus the infected. 2 days, they prefer the infected. They prefer the intact. But then once it's actually now, they avoid basically when it's when it's 2 days, they avoid the 2 days. You can see the blue is the non-infected, okay? But then if you look at 7 days, when it's infected compared to the tomato intact, they prefer the 7 days. So the question is, what is happening here? This is validation of the very first results, clearly telling you that 2 days post-infection, something is happening. So we have to come out with a way of collecting the odors below ground. What is it that the roots are releasing when non-infected and infected? And this is a system that we came up with. The same thing, what you see the black thing over there at the top, it's actually carbon. It's charcoal filter to prevent above ground odors going below ground to give us a different type of results. So that serves as an absorbent of anything that is coming from above ground. So we have a probe which goes to the soil, and then attached to it is an absorbent, which is the super Q over there, with a vacuum system, with a flow meter, which allows you to pull the odors very slowly to capture the odors from below ground. Now, one plant is not going to give you that much. In real life, in the field, you're going to get a lot of plants. So you have to actually simulate this. We did all this kind of work and found that if we have 5 plants in there, we are able to capture enough odors to simulate what is being detected by the root knot nematode. So here is the chemistry for non-infected or intact plant, and then 2 days post-infection and 7 days post-infection. This is a chemistry analyzed by gas chromatography, mass spectrometry. We identified all these. The numbers you see there are actually identifications of compounds. This is what is interesting about it. You see these are monoterpenes, and then there's one sesquiterpene, which is just around 21. These are all telling you changes. If you look at these numbers and you go through, you see changes. But the biggest change actually came from here. This compound, methyl salicylate. Now, we just plotted the amount. So you can see uninfected, which is the NI. Look at the amounts when it's intact. But once it's 2 days post-infection, the methyl salicylate level goes down significantly, like 5-fold less. And then 7 days later, it pops up again. Okay? So that gives you an indication that this must be an important compound in terms of the behavior of the root knot nematode. How about this? What else now pops up in the 2 days? Why is the 2-day post-infection avoided by the root knot nematode? Next one that pops up in the 2 days is actually methyl dihydrojasmonate. This is very interesting if you understand plant physiology. Methyl salicylate actually kicks in from what we call the salicylic acid. These are hormones, these are plant hormones, and they are derivatives of the plant hormone. For plant defense, you have to have a pathway, and that is actually either the salicylic acid pathway or the jasmonate pathway. Where the salicylic acid pathway in the intact plant and it's actually releasing methyl salicylate into the soil, the root knot nematode eavesdrops into it, to tell you that it tells it that this is a healthy plant and I can actually infect. But the moment it infects, the pathway switches now to the jasmonate pathway, which is different, to release this byproduct, which is methyl dihydrojasmonate. So the root knot nematode is able to detect the presence of that and associate it to plant defense, okay? And for that matter, avoids it. So this is really very interesting, basically looking at the quantities. But again, I said behavior drives everything. So what is the response to methyl salicylate and methyl dihydrojasmonate? That's the next question that root has to ask itself. And here we are. Here's the response to methyl salicylate. They respond almost about 80%. The root knot nematodes and Belidogyne, javanica or incognita, their response at different doses is really very high. It just tells you they're so sensitive to detecting the presence of this in the soil, and then it allows them to infect. But then if you do jasmonate, you look at even at a very low dose, you do get a very strong avoidance. So they understand basically their response to the plant is based on plant defense, depending upon which pathway kicks in, whether it's beta salicylate pathway, which they love, but avoid when the jasmonate kicks in. So this is really very important for us that at the end of the day, we've been able to understand olfactory, cues that interact or mediate this interaction. But that's not the end of it. We have to ask the next question. Is that all? Because root exudates should have more than simply just volatiles. What about non-volatiles? Here's a method to collect the non-volatiles. Take the plants out of the soil without breaking the roots gently and then put them in water for like 48 hours. You get the root exudate. We freeze dry and get the material, solid material, and then redissolve. And then you can actually carry out bioacid. We came out with this very simple bioacid. It's just a choice acid. And now investigate chemotaxis. That is the J2s, because they have to move. We introduce right in the middle, and then you have one side, it's also a choice essay. Either have the exudate or the compound against the control. So here we are with this kind of acid. These are the compounds that we actually identified. Here is the trace. This is the chemical trace, which is now HPLC because this is now non-volatile. We cannot analyze by GCMS. And indeed, this is liquid chromatography mass spectrometry. So it allows us to be able to identify compounds at the same time. We identify this phytohormone, zeatin. And why we have it green is because in bioacids, the root knot nematodes respond to it very strongly, zeatin. But look at this. These are flavonoids, luteolin, quercetin. After fractionating these, and collecting different fractions, we ended up with flavonoids in one. You have the phytohormone, and then another set of compounds where I have actually marked red. These are aglycones, glycoalkaloids. These are also associated with solanaceae. The red indicates that the root knot nematodes avoid these compounds, but not the phytohormone. So you see the link. Even for volatiles, they're using the byproducts of the hormones, salicylic acid, jasmonic acid, okay, sends a different message. And again, when it comes to non-volatiles, it's the phytohormone zeatin. So there's that kind of link between volatiles and non-volatiles. They're using the same signals, okay, to detect the presence of. So what this means is that from a practical standpoint, if you have a plant that is releasing a lot of zeatin, it is going to be susceptible. These are biomarkers in a sense. If you have a plant that is releasing a lot of methyl salicylate and zeatin, then it is going to be susceptible to root knot nematodes. If it's, on the other hand, producing a lot of these others, luteolin, quercetin, somatidine, and related compounds, then you are more likely to suppress the presence of root knot nematodes. Alternatively, if you have a plant that doesn't have these compounds, but a different set of compounds, then we must try and understand what's going on there. But potentially, if these biomarkers are not there, you likely have a plant that is going to suppress root knot nematodes. So we found this really very exciting to now say that if you have beta salicylate, which is volatile, zeatin, which is not non-volatile, but they are all associated with hormones, plant hormones, then you get attraction to root knot nematodes. On the other hand, when you have beta dihydrojasmonate, which is also a plant hormone, but from a different pathway, and then these compounds, which are non-volatile, you are likely now to be able to suppress. So that's our summary. What does this all mean from the point of view of the small-scale farmer within the African context? These are their traditional methods to suppress root knot nematodes: crop rotation, organic soil amendments, sometimes intercrop, most of the time they're not really sure about what they're doing, or come out with dead-end trap crops, or they use processed plant parts to control, apply to the soil and see what works. So sometimes they have a mixture of all these. So if we have findings like this, it means that we either come with a crop or a plant that they can use for crop rotation to suppress because they're producing a lot more of those compounds, or one of these. Our small scale farmers usually love to intercrop. So we decided that let's target intercropping. We find a plant that they can intercrop, but at the same time, it's also useful. It's either a food or it can allow them to generate income, right? So here we are. These are some of the plants they intercrop for various things. They all belong to the same class, the same family. These are Asteraceae plants. Biotechnology spilosa, it's a vegetable for certain communities, but in other communities, it's a weed. Okay. Then you have Artemisia annua, which is actually a malaria. It has an anti-malarial property. Pyrethrum, very useful insecticide, soft insecticide. Tagetes finito, marigold. Okay. So these are all, they have economic benefits, but they belong to the same family. And it became interesting for us to now screen this. Now, all you see at the bottom is, if you take the plant in the choice assay against sand in the top one, all the red, all these plants are avoided. They prefer sand. On the other hand, if you have these plants versus tomato, the green indicates that they prefer tomato. They still avoid that plants, all of them individually. The bottom one, if you now take these plants plus tomato, you put them together within the same chamber against sun. Look at that. The presence of any of these plants causes them to avoid tomato and move towards the sun. So this is really very interesting to tell you that it's the same family of plants, the same, you have different species, but they're all behaving the same way. And it makes a lot of sense from a chemotaxonomy point of view that these plants, the roots may be releasing similar components. Now, we couldn't do an in-depth study into all of this, but we selected Biotechnology spilosa to do an in-depth study because it's very easy to work with. The beauty about it is, like I said, it's either a vegetable for some communities or a weed, but the other thing is that it also serves as a trap for aphids. So if a farmer is using it, it's a dead-end trap. It can attract aphids from the tomatoes. The aphids will not do very well. At the same time, it can also suppress root knot nematodes. So these biases indicate different treatments, and we ended up having to identify these compounds from the root exudates of it. You see, they are very different from Mithra salicylis. They don't. When you look at the profile, these are the compounds that come up, and actually, basically, in the presence of, they mask the bioactivity of tomato root. They mask the activity of Mithra salicylis. So that gave us some information about. But what about the non-volatiles? The volatiles work. So we set up this experiment that was all done by Ruth. Put the two plants together, and then also look at the exudate that comes from Biotechnology spilosa and pass it over onto tomato. That's the one you find on the right. Okay. So you water the plants, and then the exudate ends up going into the tomato. Look at the data. When you just have the intact plants and you look at the number of galls, you see when you infect on the left, you get a very high number of galls. But once you intercrop with Biotechnology spilosa, the number of gall goes down. That's the intercrop. Even with the drip, it just tells you it goes down. But the drip is not as effective as the actual plant. So it means that we are losing certain components from the drip, but it's still very effective. It tells us that if you take the root exudate, you will actually now identify at least a certain percentage of the components responsible for this. We asked ourselves, is this really true? And the way to validate this is to take another Solanaceae crop, which we did with nightshades, 9 shades, okay, eggplant, I repeated this experiment, the same thing, intercrop and the drip, and look at the data. So any solacinic crop you do with Bidens silosa or any of the asteraceae plants, you're going to get this kind of. So it tells us that this is actually a very useful plant. Here's the chemistry of the profile of the exudate. You get all these compounds, but don't worry about the chemistry. Very simple ones. I'm just going to focus on these ones. There's ascorbic acid, nicotinic acid, different classes of compounds, phenylalanine, tyrosine, these are amino acids. Then you have hydroxybenzene, camphorol, it's a flavonoid, and so on. But why we are focusing on this is because there's something very interesting associated with this. We biocide all these. If you look at ascorbic acid and nicotinic acid, look at the top. Percentage inhibition in Hegachi. They actually suppress Hegachi. But then when you begin to look at mortality, blue is actually now ascorbic acid. So it tells us that the most potent that Bidens pilosa is releasing within its root exudate that is causing suppression and is non-volatile. It's actually a vitamin. That is what is causing that suppression. Apart from the volatiles, this is so different from the Solanaceae, which was giving us solacidine and tomatidine as a different class of compounds. So that's the beauty about this finding. So what we can summarize here is that if you have tomato or any of these Solanaceae crops for the small scale farmer, what you can recommend to suppress root knot nematode, it's actually an asteraceae plant like Bidens or you can Artemisia annua or some of the Pyrethrum. So they'll do the same thing, the same trick. Ruth, like I told you, was a shining star. Look at all the publications that came out from Ruth's work. She did a great job. And our favorite journal was Journal of Agri and Food Chemistry, which is one of the prestigious journals of the American Chemical Society. She published quite a lot in there, and she actually received a lot of prizes as well, including contributing to an ACM Symposium series book chapter on roles of natural products of rational pesticides in agriculture. Very exciting to work with such, a young lady. The final one I'm going to talk about is now the potato cyst nematode, which I talked about last time on Tuesday. So I'm going to basically summarize that. And that's where we have this young lady, Julia Tuchola, coming. And she also came out with very simple biases. Does it hatch or not? It's basically a yes or no thing. She was able to demonstrate hatching with the root exudate. So the question is like, now what is the chemistry? What is in there? So many compounds against similar classes. Very interesting. Amino acids, look at zeatin. There's also dimethyl dihydrogesphosphate. This is potato, but you see it's Solanaceae. So we are not surprised to be seeing similar solanoclepin A, alpha chaconine, these are glycoalkaloids, which you also find in tomato. Okay. So what did she do? She now had to test these compounds. It's a little busy, but I'm going to make it simple for you. What is interesting is that what you have above actually supports emergence, below doesn't. And interestingly, what supports emergence here for the potato cyst nematode, solanidine, apart from what is known, alpha chaconine and others which supported tomatidine. Why I have actually put a box around this is because these compounds were actually suppressing root knot nematode, host finding, but it's the reverse for potato cyst nematode. When you get the signal, they actually hatch. So it tells you they evolved differently, total two different species, and they're using the same system, but in a different way. Look at this. I've also zeatic, dimethyl. They care less about the plant hormones. They're more interested in the other ones which suppress root knot nematode host finding. So you get that kind of interplay, but in a different form. When it comes to look at which ones were insisted or not, it's the same thing. You get them emerged, but they were insisted. It tells you that the signal was not enough. They did a blend of some of these compounds, like in the root exudate. And then it's the same compounds which you find insisted. And then in the case of zeaxanthin and betadine, they don't really care so much about it. What Juliet did was to look at additional components, and this is work we've not published. I was telling Laura the other day. These are all additional compounds we've identified from the root exudate. And interestingly, some of them are able to give us some emergence, certain level of emergence. What is interesting here, some of these acids, look at salicylic acid. You look at salicylic acid, it gives you a little bit of emergence, which is actually one of the pathways of the plant hormone signaling. But most of these are derivatives which actually also contribute to that. But here is what is even more interesting. What suppresses root knot nematode post finding as a non-volatile? Here we are, ascorbic acid. Look at that. It's a post emergence of potato cyst nematode. Very interesting indeed. And again, when you look at whether they actually did eventually emerge or not, most of them were insistent, which tells you the signals were not particularly great. But more importantly, how do we now utilize this information in crop rotation for, again, we can actually think about intercropping, crop rotation, or whatever. But small scale farmers in Africa, because it's an invasive species, they don't know what to do. So the solution now came from colleagues from North Carolina State University, collaborating with the IITA, and then of course, colleagues from ECP, where I was involved, with this technology, which is the wrap and plant technology, where banana paper was made by bioengineering the paper from the banana rachis, which is a waste material, and then you wrap the seed and plant. And this is a highlight of it. The white indicates where it's the control, where you don't have the paper, and then the red indicates where you have paper. You can clearly see it. The yields are this 3 to 5 times when the paper is impregnated with 1,000-fold less ivermectin compared to where you just have the paper itself without ivermectin. You still get yield about 3 times. But when it's ivermectin, you do about 5 times. And then a decline in cyst population in the soil, you can also see whether it is impregnated with ivermectin or not, you will get a decline in. So even the control paper works beautifully. Now, what does this mean? The paper is biodegradable. So this is just to show you that as the potato grows, the paper also breaks down. So it doesn't actually leave residues. And by impregnating a very low dose of the ivermectin or nematicide, is also to keep it within the soil and by the root environment and not spread. Okay. So it breaks down. But then if you take the paper, we can always recover the hatching stimulants. What happened to the hatching stimulants? These are the hatching stimulants which have been identified for like almost 2-3 decades, close to, more than 2 decades. People are still working on this. But then it's actually solanoclapin A, which is the tetranortriterpenoid. And then these other components, which are glycoalkaloids, alpha-solanin and chaconine. These are compounds that root knot nematodes avoid, but the relatives actually are preferred by potato cyst nematode. To do this kind of work, we have to look at what happens to the paper, whether impregnated or not. We start from the top. That is the root exudate signal, identifying these components. If you go to the bottom with the paper, we don't find these components. On the left, cellulose is part of paper, and then lignin. We just use cellulose. When you just pass the root exudate over cellulose, they also disappear. So the question is, what is happening? It's actually an adsorption. And then to prove the adsorption on the right side, we extracted the paper or the cellulose with solvent, and we were able to recover the same compounds, which tells you that we're just bound to the paper. So to summarize how the paper works, on the left side clearly tells you that in the absence of the paper, the potato cyst nematode is able to complete its life cycle and infect the potato roots. But on the right side, once you have the paper, what happens is that there are three modes of defense. The first one is that the paper serves as a physical barrier. It doesn't allow the J2s to penetrate. The second one is that the presence of the nematicide serves as a killing agent, but just within the root area. But in the absence of the nematicide, some still die. But more importantly, is the bottom one, where it serves as an adsorption. It's like a sponge that absorbs the chemical signals useful for completing the life cycle of the potato system. So the science behind it, if it's adsorption, is because cellulose is actually full of sugar molecules. And then if you take one hatching factor, which is the bottom solar nucleotide, then it means you just have hydrogen bonding. And that's the very reason why we can easily recover by extraction and analysis. The young lady also did very well and published quite a number of papers, one in Frontiers in Plant Science, and then the other one in Nature Sustainability. Some of the authors are here, Onesmus, Flora, and then there's also a review paper in Pest Management Science. It's really great. So to conclude this whole presentation, Laura is right there, working with farmers, is that chemical ecology of plant parasitic nematodes reveals opposing differential modulation in chemical signaling in root knot nematodes and potato cyst nematodes. I've just explained to you that what root knot nematodes like are not liked by PCA and vice versa. And then RKN is a generalist, it's polyphagous. So it makes a lot of sense for it to eavesdrop into a healthy plant that is releasing methyl salicylate or zeatin. It just tells you that once the phytohormone is there, the plant is healthy. And for that matter, it can infect. On the other hand, PCN is more specific. It has to have the right signal to hatch, and then you can have the J2s. That is the very reason why you can stay in the soil for 20 to 30 years and plant other things. So long as the signal is not there, because of their specificity, it's not going to hatch. And then for small-scale farmers, we're teaching them that science-based evidence provides opportunities to developing low-cost, eco-responsive solutions to manage plant parasitic nematodes in their smallholder farm. And like I said, despite all the science, we have to simplify it for them. Either find a plant source for some of these compounds, or come up with some sort of formulation they can use to work with. I'd like to acknowledge contributors to this presentation from different institutions, CPI, TA, where the students were registered, Kenyatta University, USDA, North Carolina State University, the great team there, and the supporters, Bill and Melinda Gates Foundation, CEDA, and all these others. And ECP has a consortium of donors. They support us in many ways, and we must thank them. And finally, without ICP and of course VID, I wouldn't be here. Thank you all for your attention.
So many questions, only make a remark that it's a fantastic opening of an whole new field that people don't realize. One is the field of the volatiles itself, how you can study them and how important they are in the plant field, and also already on the way the compounds are exudated. Now one is completely sure that food, that's not something that is material, it's a network of nanotubes. And the nanotubes 10⁻⁹, then the hydrogen bonds that you pointed out, play completely other role. And so at the moment, there is a lot of analysis. If you be able to do the analysis, and in a certain way, the natural products were studied again in the late 40s, early 50s, because there was paper chromatography.
Yes.
But that's the separation. It's cellulose, and you do it again in your systems, and you will find many compounds. Then molecular biologists can come in the pathways, and you will find better. So everybody will be happy, and at least people will trust and will fund the systems that you do if you can improve the rationality of the molecular biology that is behind it.
Yeah.
You can already use it in a cheap way and improve that. So it's a very crucial point, I think, in general, and also in knowing plant physiology that is so recent also coming up. There were some descriptions, but the whole rationals for each of the steps that can be found. And then when they see it is like that, more young people will follow it and will do it. So it's extremely important. Thank you very much.
Thank you so much for that comment. I really appreciate that because I didn't have that much time. I basically finished right on time, but the future outlook is what is just outlined, because what I've provided is actually the chemical signaling, but there has to be a molecular basis for that. What kind of genes are expressed, in terms of defense? So we have to understand the molecular aspects also of that, get into the genetic aspects of it. And then, because we're working within the rhizosphere, what is happening to the soil microbiome? Where do they come in? And then also in terms of the plant physiology, all right, particularly in the case of the wrapping plant technology. When we use the paper, how is it influencing the microbiome within the soil? What is the response of the plant in terms of its agronomic characteristics, in terms of its physiology, in terms of nutrient uptake, and so on? So this is just one small component of a bigger picture, and that is the very reason why multidisciplinary approaches to doing things actually gives you a better feel of all the interactions and so on. This is just basically looking at chemical interactions, but yet there has to be the molecular aspects of it. And of course, understanding what kind of genes are upregulated or downregulated in response to all these interactions. So thank you very much for that kind of comment. Thank you very much. Say that again. Yeah, there's that possibility there, because you do have plant-plant communication, okay? And so whatever we've done, it's actually lab-based. That is the very reason why it's always important, even validating in the lab, you need to go to the field, because that is now going to tell you if you do have another plant intercropped. An intercrop doesn't mean necessarily putting it next to each other. There has to be some spacing, and decide what kind of spacing is going to lead to that suppression and all that kind of thing. So we need that kind of information by an agronomist to tell us all that sort of. So plant-plant chemical communication is also important. It might reinforce above ground interactions in terms of maybe not bringing in pests or maybe introducing more pests, or perhaps even promoting natural enemies to come in and that sort of thing. So it basically opens up new avenues for looking at various interactions. Yeah. Good.
To take up the first, you can with the single compounds that you see, if you absorb them in charcoal, because charcoal, the nanopores are still there, but you kill all the living material, and so then you can apply really in an original way that, because the water flow to that is another story, because that's the way for in arid soil keep water.
Mm-hmm.
Will not touch that. But if that moment, I forgot why what I was, but what I was saying, pointing out for reaching up. There's one part is the science, what really happens, but in society, it's the social economy that counts.
Yeah.
How the people can survive, how the big business can make money, and how the environment can be kept. And then you have to see the different methods, and you only have the cheap methods of co-cultivation,
Yeah.
where you do it with the maximum yield on the based on the molecular science that you what you do better. And then you have the compounds themselves, and that's then for the chemical industry to see what they will do. But that are the sponsors at the moment, and until the society develops better and has a better interaction between the sponsors and the people who are doing the living.
Yeah, yeah. If I may just comment, thank you very much for that as well, and also for what you've asked about, because our target stakeholders are smallholder farmers. This is the very reason why we always have to look at technologies that are very simple for them, but it still takes some time, but a lot shorter than when you are doing biotransformation. For instance, I mean, we know the secondary metabolites, and then if we know the genes, and so you can actually introduce the gene into that specific plant. But we all know that it's not an overnight thing, So that's a longer process, but that's a lot more science, okay? And you might perhaps end up with a product which, at the end of the day, he was talking about social scientists. You might end up with a product which is resistant, against plant parasitic nematodes, irrespective of whether it's root knot nematodes or maybe broad spectrum for both root knot nematodes and cyst nematodes. But then the harvest, the yield, which is going to come out of it, they may not like the taste. You know what I mean? You see, there are many factors involved. So what they have, that is what we work with. This is what they like. We find a solution that fits into their traditional practices and things like that. Because if you take a lot of some of the institutions set up many, many years ago, they are in breeding, okay, including IITA, IITA, with the breed, a lot of legumes, beans, and cowpeas and so on. They have thousands of cultivars. And not all of them are out there, just a few of them, because there's this socio-economic aspect of it where farmers may say they don't like the color, even though they get a lot of yield, or they don't like the taste, that kind of thing. So, and then it varies from community to community, and from one community to the other. It's very challenging. So it's best to have something that you can do great science and all that, but you have to package it to fit the stakeholder, your target group.
Okay, so Xavier Cheseto, that I think
I know who he is very well. He's from my department.
It's saying, thank you, Professor, for this very informative talk. Did you have an opportunity of evaluating the effect of the paper and the pesticide on the soil microbiome and soil health in general?
Yeah, I mean, these are all follow-up studies. It's important, even though the nematicide is a thousand-fold less than the conventional use. it's always important to collect soil material and find out how much is it there as a residue. So those are follow-up studies, which at the moment, no. What I can tell you is that the paper actually now is being used for other tubers, okay? And those who are doing it in the field, our IT collaborators are very excited about it, but they may need to begin to look at residues in the soil, even if it's within just the root environment and things like that, or even uptake into the roots, okay? And is that contributing to some of the dynamics that we're seeing in terms of the physiology of the plant and things of yield or so? We don't know. But the levels are so low that I doubt whether they'll be systemic in terms of uptake. Yeah. Yeah. Yeah, 1,000-fold less. So that's really very little. But it's a good question, and scientifically, it's always good to have the data to say it's yes or no. Okay. All right. Thank you.
Able to dissolve in something, and when you compare it with your stage, which kind of maps do you connect with?
I think Laura can answer that, because they do a lot of field work with the farmers and so on. But I think that what I can say is that most of our farmers, when you're doing crop rotation, because their land sizes are very small, today they may plant maize, tomorrow it's something else, and that sort of thing, and so on. So whether they are actually plants, maybe okra or whatever, I have no idea. But it's always good to get that kind of information from farmers because it is a two-way exchange. It's not always us teaching them. We also learn from them a lot. So, but I don't know where Laura you can contribute to that. Mm-hmm.
With that, several times, get applause, part of this, they really go and the best you see to the new I said that because where I stay in which I think it's national and state institute. We experience that because once we know that by planting amaranthus, for instance, when we go of some especially the people after like 1-2 cycles, we put amaranthus there, you do the soil other. So it was giving us.
But for what? Which pest? Which pest? Nematode.
Yes.
Okay. But it must be root knot nematodes, not PCN, not cis nematodes, root knot nematodes. Yeah, yeah.
There is an industry about pop and seeds that is the new value chain, which is others think somewhere like benefit and the money again at one and that is it's not what it is. So something that you give this technology, awareness, show them and see what downstream of the value chain. This is has not come to what it doesn't know if that doesn't
Okay, I think I continue to be around for a while, so we will be much to have an last question, but I think we can just run that discussion.
The sticking.